Hello everyone, this is Dr. Vishal Trivedi from Department of Biosciences and Bioengineering, IIT Guwahati. And in the course Enzyme Science and Technology, we are discussing about the different aspects of enzyme and how the enzymes are doing the different types of functions and catalyzing the different types of reactions. So in this context, so far what we have discussed about the history of the development of the field of enzymology. Subsequent to that, we have also discussed about the enzyme nomenclatures, and then we also discuss about the enzyme classifications. And in the current module, we are focusing more on the enzyme structures, right? Because the structure play a very crucial role in identifying the substrates and then catalyzing the reactions and then generating the products. And then once the product is being generated, it is going to be released from the active site. So when we were discussing about the enzyme structure in the previous lectures, we have discussed about the primary structures, we have discussed about the secondary structures. So in today's lecture, we are going to discuss about the tertiary structures and how you can be able to use the different techniques to solve the tertiary structures. So let's start the today's lecture. So what we were discussing about the protein structures, and what we have discussed so far is that the protein is made up of the 20 naturally occurring amino acids, and a typical protein amino acid is containing the two component, amino component and a carboxyl group, and that is attached to the central alpha carbon. And what you see here is that the C alpha carbon, on that you have the amino group on one side and the carboxyl group on the other side. And then it also has the side chains, and based on the side chains you can have the 20 different types of amino acids. So what you see here is the side chain of the different amino acids, and as you can see, some are positively charged amino acids, some are negatively charged amino acids, some are hydrophobic, some are hydrophilic, some are polar groups and all that. So since all these amino acids will come and join together with the help of the peptide bond, that is constituting the primary structure. So what you see here is that the primary structure of a protein is defined as the amino acid sequence from the N to the C terminus with a length of the several hundred amino acids. And the primary structure is going to be folded into the secondary structure, and then secondary structure is going to be folded into a tertiary structure. So within the secondary structure we have the alpha helix, we have the beta sheets, and we have the turns, right? So with the help of the turns, the alpha helix and the beta sheets can actually be able to form the different types of secondary structures or super secondary structures, and these secondary and the super secondary structures actually can come together and they will give you a tertiary structures. So in today's lecture we will see how we can be able to solve the tertiary structures. So in the tertiary structure, the secondary structures are going to fold to give the higher order organization, commonly known as the tertiary structures, whereas the quaternary structure is going to be present if the multiple polypeptides are involved in the constitution of the protein, and the tertiary structure of these different polypeptide chain come together to form the quaternary structures. Now when we talk about the tertiary structures, the tertiary structure is a very complicated structure of the protein, where the many of the secondary structure and as well as the super secondary structures are coming together and giving rise due to tertiary structures. So when we talk about the protein structure determinations or the tertiary structure determinations, we have first question is that how we are going to perform the protein structure determinations. So we have two options, option 1 and the option 2. So option 2 is more of the experimental method for which you are interested to solve the three-dimensional structures, and you have the two pathways. One is this pathway, which is going to be called as 2A, and this pathway is going to be called as 2B. So this pathway, what you are going to have is you are going to this is called as the X-ray crystallography, and in this the major pathway is that you are going to generate a crystal of this particular protein, whereas in the case of NMR spectroscopy, so you have two options, experimental options, which you can use to solve the protein structure. One is the X-ray crystallography, the other is NMR spectroscopy. In the X-ray crystallography, you might have to generate the crystals of that particular protein, and then these crystals can be studied in the X-ray crystallography, and that is going to give you the protein structures. Similarly, in the case of NMR spectroscopy, you can express the proteins and then you can actually be able to record the NMR spectra and you can be able to solve the structure of the protein with the help of the NMR spectroscopy. So, both of these experimental methods are going to give you the protein structures. So, what we are going to do is we are going to first discuss about the experimental method and then we are actually going to focus about the non-experimental method. So, in the non-experimental method, these are also called as the computational method. You are actually going to use the protein sequence and that you are going to use different computational tool to do the molecular modeling. So, what we are going to do is first going to discuss about the experimental procedures and then we are going to discuss about the non-experimental procedure. When I say non-experimental means I am talking about the computational approaches. Now, as far as the experimental approaches is concerned, once you have the proteins, you can actually be able to generate either the crystal or you can express the protein and that will give you the NMR spectroscopy. So, you have two options, 2A that is the X-ray crystallography or 2B that is the NMR spectroscopy, and both of these are actually going to give you protein structures. So, let us start with the crystallography. So, first question is what is crystallography, and before we discuss any of these methods, I just want to make it sure that we are not going to extensively going to deal either of these methods, either the X-ray crystallography or NMR spectroscopy. The purpose of this today's lecture is that we are going to very briefly going to tell you about the X-ray crystallography or as well as the NMR spectroscopy, so that you will be able to follow the content and you will be able to understand the potential of these techniques. If you are interested, you want to explore some of these techniques, then it's better to go with the well developed MOOCs courses where they have actually discussed about the X-ray crystallography. So, these topics are relatively big, so that cannot be covered in a 1 or 2 lectures. So, what is X-ray crystallography? X-ray crystallography is a form of very high resolution microscopy, so that you can be able to see the crystal and you can be able to see the protein structure inside, right? Just like as we actually see the slides under the normal light microscope, you can also be able to see the protein structures if you have the tools, and that is what the tool is, X-ray crystallography. So, X-ray crystallography is a very high resolution microscopy which actually will allow you or enable you to see the protein structures. So, what you see the potential, right? What you see here is the potential of different types of microscopy. You have the light microscopy. So, light microscopy is going to use the light, which is actually the length of the 300 nanometers, and that actually is going to give you the visualization of the cells or the subcellular structures with the help of the phase contrast. Whereas, when you are going to talk about the electrons, so you can actually be able to have the electron microscopy, and the wavelength what you are going to use is 10 nanometer. So, light source what you are going to use is 10 nanometer, and that is actually going to allow you to see the cellular structure and as well as the shape of the larger protein molecules. In some cases you can be able to see the DNA structures and you can be able to do all those kind. Whereas, when we use the X-ray as the illumination source, the wavelength is going to be 0.1 nanometer, and it is actually going to be allow you to see the atomic details of the protein. So, the difference between the electron microscopy versus X-ray is that electron microscopy is only going to give you a gross structure of the proteins, whereas the X-ray crystallography is going to be even finer, and it is actually going to give you the atomic details of the proteins, and that is basically because the wavelength what they are using to visualize the objects, right? In the case of electron microscopy, it is going to be 10 nanometer, whereas in the case of X-ray crystallography, it is going to be 0.1 nanometer. So, since it is a form of the high resolution microscopy, it enables us to visualize the protein structure at the atomic level, which means you can be able to very precisely be able to see the different atoms. What is the principle of the X-ray crystallography? So, it is based on the fact that X-rays are diffracted by the crystals, right? What is the difference between the conventional microscopy versus X-ray crystallography is that here there is no lens involved, which means you cannot just use the X-ray crystallography and visualize the object just like as we are visualizing the cells or the subcellular structures in the slide. So, if you are interested more about the studying the X-ray crystallography, you can be able to go through with this particular articles. Now, what are the different steps you can be able to use in doing the X-ray crystallography? So, in the step 1, you are going to first express the proteins. So, majority of the time when you are actually going to do the X-ray crystallography, it is going to be an overexpression of the protein and overexpression and purifications. And that is very important, because if otherwise you are not going to achieve the desired amount of the protein, and that's how it is actually going to be a problem for getting the crystal. Then the step 2 is going to be you have to do a protein crystallization. So, that is actually going to give you the crystals, right? That is what is the prime requirement of the X-ray crystallography. Then the step 3 is going to be diffraction data collections, right, that you are going to use with the help of the X-ray diffractometers. And then step 4, you are going to get the diffraction data, and then based on that you are actually going to generate the electron density map, and you are going to do the structure solution with the help of the computational programs. And then step 5, you are going to use the refinements, and you are going to do a model building, so that you are going to get the protein structures. Now, this is what I was talking about. So, there is an excellent MOOCs course which is available on the NPTEL website, and that is actually you can be able to take. So, if you are interested to understand all of these steps in detail, right, because here we are just briefly overview, we are giving you an overview. So, you can actually be able to go through with this particular NPTEL course, and that will actually going to give you the detailed insight about each of these steps. Now, let us talk about the first step. So, first step is the overexpression and purifications. So, in this you are going to do the cloning of the protein into the suitable vector, right. So, cloning of the protein or enzyme in the vector. Then you are actually going to do the transformation. So, once you do the transformation, you are going to get the transformed colonies, transform bacteria, or whatever the expressions host you are going to do. This all we are going to discuss when we are going to discuss about the production of the enzyme. Transform bacteria, and then that is if you do the induction, right. So, that is going to give you the protein, and then ultimately you are actually going to do the protein purifications, because protein purification is required, because you are supposed to have the 99% pure protein for crystallizations. In the step 2, you are going to do the protein crystallization, so that you can be able to develop a crystal, and that crystal is actually going to be diffract to X-ray, and that's how that going to give you the diffraction data. Now, let us talk about the crystallization, the step 2, right. So, first question is how the crystallization works, right. I am sure if you want to understand the process of crystallization, what you can do is, or you can even do that in your home also, do one thing, right. You take a glass, right, and just add small amount of sugar. So, for example, you take 10 grams of sugar, right. So, that is going to be there, right, or you can use some amount of urea. right? So these are the solutions, right? So you prepare the solution and let this beaker, this glass to be remain open. So if you take the steel glass and let it be open, so what will happen after some time is that all the water what you have added into this is going to evaporate. So after the evaporation, what you are going to see is that in the glass you will see the spikes, you will see the spikes type of crystals, and that these are the actually the crystals. So you, when you take the sugar, it will form the sugar crystal, whereas if you take the urea, it is actually going to give you the spike shaped crystals. So how it is actually happening? It is happening because when you are evaporating the water, you are basically increasing the concentration of the solute, right? So you are increasing the concentration of the solute, right? And in that process, what will happen is that the molecules are going to come together. So for example, you have one molecule like this, the other molecule is very far away, but when the water molecules, so water molecules are present in between, right? So once after evaporation, what will happen is these two molecules will come together, right? And you can imagine that many such molecules, when they will come together, eventually that is going to give you the crystal, right? These crystals could be produced for the inorganic molecules or organic molecules, or these crystals can be produced for the protein. So under what condition the molecule or the solute is going to give you the crystal? When the similar kind of proteins, like similar kind of molecules, are going to come together and will give you a homogeneous stacking. So if there will be a homogeneous stacking, they will actually going to come together and will give you the crystal. If it is a heterogeneous stacking, for example, first molecule is going to be placed like this, the second molecule is going to be placed like this. So in those case, they will come together, still they will come together, right? So you can see still they will come together, right? And they will stick to each other, because as soon as you remove the water molecule, they will come together, they will have no option. But in this case, they will not going to give you the crystal. Instead, they are actually going to give you the amorphous salt, okay, which means they will give you a precipitate, or they will give you the amorphous salt, and that is actually not going to diffract. So they will not going to diffract to the X-ray, whereas a crystal is going to diffract. So what I am trying to explain is that when you are actually going to bring the molecules together, they will either be present in the same conformation or they will be present in the different conformations. So when they will be present in the same conformation, they will stick to each other, they will achieve the lowest energy, and that's how they will going to form the crystal. But if they will be in different orientations or different conformations, they will come together, but they will going to form the amorphous powder, and that amorphous powder is not going to diffract. Why it will not diffract? Because the diffraction pattern from this conformation and diffraction pattern from this conformation is going to be opposite. So if we can imagine that if I got the positive diffraction with this guy and negative diffraction by this guy, so ultimately I am going to get the nil diffraction when these molecules are going to be present in the amorphous salt. The other issue why the crystal is also diffracting is because it actually keep all the all the protein molecules or inorganic molecules in the same orientation. So you are actually going to get a positive diffraction. Now when we talk about the crystals, so there is a defined size and there is a defined clarity, there is a defined parameters which are actually need to be followed. So when we produce the crystals, they are supposed to be small in size, which means they should be of 1 mm in size. Then they should not have any defects, okay. So which means when I said they should be of diffraction quality, which means they should not have any kind of defects. What are the defects? You should not have any cracks, it should not have any air bubbles, or it should not have the mosaic, okay, which means mosaic means heterogeneous nature of the proteins present in the particular crystals. Now, when we talk about the methods of crystallization, so there are tons of methods what you can actually be able to use for producing the protein crystals. But what we are going to discuss in this particular lecture is the hanging drop method and actually also the sitting drop methods. So let's first talk about the hanging drop method. Okay, so in a hanging drop method, what are the material you require? You require a cover slip, you require a 24 well dish, and you also require the mother liquor. Okay, now how this hanging drop method works is that you are going to have the well, right? You're going to have the well. On top of this, you are going to have, and fourth is you also require the protein solution. So this protein solution is purified. Okay. So what you are going to do is you are going to have the a well, right? And on top of this well, you are going to have the cover slips. Okay. And on this cover slip, you first what you are going to do is you are going to have a cover slip. On this cover slip, you are going to make the drop of the proteins. Okay, so you're going to make a 4 microliter drop of the protein molecules, right? And then you are going to add the 4 microliter of the precipitant, right? Or I will say mother liquor. Okay. So then you mix them together and make a drop on this cover slip, and then you actually invert this, invert in such a way that the drop should face the well. Okay. So what will happen is you will see the well drop is going to be hang like this. Okay. And then you are going to seal this with the help of a vacuum grease, and then ultimately it is going to be a isolated chamber, and then you keep it, and then you can observe the crystallization apparatus or crystallization setup with the help of the microscopes. Now what will happen is that eventually the water is going to come out. So this well is actually being filled with the same precipitant what you have added into this protein molecule. So you are going to have the precipitant into this. Okay. So what will happen is that when you do this, the water molecule is actually going to be taken up by this downstream liquid, right? Because the amount of precipitant is going to be on a higher side into this, right? So it is going to be 100% here, whereas it is going to be 50% in the case of the drop, right? So because of this change in the concentration of the precipitant, the water is going to come out from the drop and it will fall into the precipitants. Okay. So once it happens, it is actually going to do exactly the same what we have just now discussed in the glass, right? In the steel glass, or in the case of salt. So what will happen is, you can imagine that if you have a drop like this, right, and you have protein molecules. So what will happen is that initially the protein molecules are going to be very far away, right? But once the water will evaporate, these protein molecules are going to come together, right? And that's how they are actually going to start forming the crystals, right? Although this is not true in most of the cases, right? It's not that only the removal of water is actually going to bring the protein molecules together and you will going to be the crystal. No, it is not that easy. What is the requirement? The requirement is that the protein molecules, when they come together, they are actually should be attending the minimum energy, and they should all be attending the similar conformation, so that they will be sticking to each other and they will give rise to the crystal. So how the crystallization is going to appear? The crystallization is going to appear like this. Okay, so eventually you are first going to have the nucleation on the corner of this particular drop. So this is I am just showing you a drop. So it is actually hanging like this, right? So you can imagine that this drop is like this. So what I am showing is actually this cross section. Okay, so this cross section is what I am showing. So what will happen is, in an ideal situation, when you have added the precipitant which is stabilizing the protein structure, which is maintaining the minimum energy, and which is also removing the water at a very controlled rate, right? When you do this protein water removal at a controlled rate, you are going to induce the protein crystallizations. So what will happen is that on the corner you are actually going to have the nuclei, the protein crystals nuclei are going to be generated. Now what will happen is that this protein nuclei are going to swim into the center. So you can imagine that the crystals are, the nuclei are going to be developed here, and then they will actually going to swim into the liquid. Okay, so once they swim, they are actually going to start collecting the protein molecules. Okay, and eventually what will happen is that they are actually going to form the crystals, right? So in the second row you are going to get the crystal, and as these crystals will go in the center of the tube, which means they will actually be in the center, they will be start eating, or they will be start collecting the protein, and that's how you are actually going to get the bigger protein crystals in the center of this particular drop. This is the ideal situation under which you are going to see the crystallizations. So this is all about the hanging drop method. You can also try out the sitting drop method, because one of the major limitation of the hanging drop method is that you are supposed to hang the drop into this particular 24 well dish, right? So in that case, there is a limitation until you can actually be able to keep the protein molecules. So you cannot keep beyond certain concentrations, which means you can try out the 4 microliter versus 4 microliter, you can try out 6 versus 6. Once you increase the 6 plus 6, or even if you go beyond that, the drop will actually going to fall into the, right, it will not going to hang, right, because the surface tension of the water is going to be not allowing them to stick to the glass, right? So because you cannot have a desired concentration of the protein into this, and some proteins require the even higher concentration. So you can imagine that if you have got the nuclei, but that nuclei is not growing, that means there is a shortage of the protein in this drop. And how you going to solve that problem? You're going to solve that problem when you are going to try the sitting drop method. So let's discuss about the sitting drop method. So in a sitting drop method, what you have is you have a bridge on which you can be able to place the protein molecules, and that's how it is actually going to work. So sitting drop method, you have you're going to have the well, right, just like as we have the well in the hanging drop method. But on the hanging in the center, instead of you are going to have the cover slip, it is actually going to have a bridge like situation. Okay, so it is going to have the bridge which is actually going to have the depression, and this depression easily can take up to 50 microliter of the protein mixture. So again, you are going to fill this tube with the help of the precipitants, right, whatever the precipitant you want to try out. And then what you're going to add is, for example, if I add the protein molecule, so I can place the 25 microliter of the protein solution, and I will mix it with the 25 microliter of the precipitants. Okay, and then I am going to seal this with the help of the vacuum grease, and I will seal it with the help of the cover slip. Okay. Now what will happen is exactly the same thing will happen. So for example, if I have added the drop, right, so it is a 50 microliter drop. Earlier we were just making the 8 microliter drop. Now again the same thing will happen. The water will evaporate, right, from this drop into the liquid, and eventually the protein will concentrate, and that's how in that process it is actually going to be crystallized. So with the sitting drop method, you can be able to try the large protein molecules or large protein solution that actually is going to reduce or it will take care of the lower protein content in the drop, right. So that is actually going to increase your chances of getting the protein crystal or bigger protein crystals, right. In some cases, people also try using the nucleation. So for example, if you are trying with the hanging drop method, you are actually going to get the nuclei, right? But their nuclei are not growing. So in that case, what they do is, with the help of the pipette, they actually isolate these nuclei, right? And then they put these nuclei into another drop. Okay. So once they put these nuclei into another drop, there is a possibility that they may actually get the bigger crystals. So as I said, you can actually be able to consult or refer this particular online course. You can even register for this online course if you are interested to study more about the crystallography. And once you do the crystallization, you are actually going to get the crystal like this, right? See, these are the protein crystals, right? And this picture is being provided by the Professor Shankar Prasad Kannojia from our department only. Since he was kind enough, we actually took the liberty, and that's how we went to his lab, and some of his students actually have demonstrated how you can be able to crystallize the protein using the hanging drop method. So let's show you the video where we have discussed about the hanging drop method and how you can be able to perform the hanging drop method in a laboratory. So the material what you require for the protein crystallization is as follows. You require a 24-well dish. You require the mother liquor where you would like to test the crystallization conditions. You require a forceps, and then you also require a siliconized cover slip. So you have the two choices to prepare the siliconized cover slips. Either you can buy the siliconized cover slips from the vendors like the Hampton Research or the Sigma, or you can actually be able to prepare the siliconized cover slips by coating these cover slips with the help of the Sigma coat. So you can buy the Sigma coat from the Sigma company, and you can actually be able to dip your cover slip into the Sigma coat, and that will actually going to make the siliconized cover slips. So let's start the demo. So first thing what you have to do is you have to put a vacuum grease. So what you can do is you can just fill the vacuum grease into a syringe like this, and then you can actually be able to make the rim on top of the this actually 24-well dish, right? So that it is actually going to seal when you are going to keep the cover slips. After this, you are going to add the 1 ml of the mother liquor or the crystallization conditions. So for example, in this case, since we are trying to do the crystallization for the lysozyme, we are going to do the use the mother liquor, which is the 1.5 molar NaCl and 100 millimolar sodium acetate pH 4.6. So let's put the mother liquor, and okay, now you have to ensure that when you prepare the mother liquor, it should be filtered sterile and it should be filtered so that there should be no suspended particles. Now what you're going to do is you're going to take out one siliconized cover slips with the help of the forceps. So you can just take out the cover slip, put it into a tissue paper, right? And then you're going to make a drop of the proteins, and so let's do that. So in this particular experiment, we have taken lysozyme as a model protein. So you know that lysozyme is very easily crystallizable. So we have done is we have prepped the cover slips onto our tissue paper. Now we are going to add the protein. So you have many choices of adding the proteins. You can add 2 microliter of protein, you can add 3 microliter of protein, 4 microliter, and so on. So in this particular demo, we are putting the 3 microliter or 2 microliter of the protein. So we have made 2 microliter, right? And now we are going to add the 2 microliter of the protein. So this I have prepared the protein which is filtered also. So this is a 5 milligram solution, and I'm going to add a drop in the center of this tube cover slip. When you add the protein, you have to ensure that there is no bubble which is going to be formed in the protein solution, because the bubble is actually going to deactivate or inactivate the protein, and also it is actually going to distort. And now we are going to take the 2 microliter of mother liquor, and we are going to add that also into this protein solution. Then you are actually going to make this as 4 microliter, so that you can be able to mix both the protein as well as the mother liquor solution properly. And now your drop is ready, right? Now you take the drop into your hand, okay, and then you have to invert this like this, okay, and when you invert, you have to put this onto the top of the well, okay. Now once you have put it on the top of the well, you can use the used tip, and then you press the slide on this well very carefully from all the ends, so that it should not break the well, and it should also not have any air gaps. So when it's got sealed, initially what you have to do is you have to cover it with the lid, and then you have to observe this drop under the microscope, and you have to observe to see that there is no precipitate which is going to be formed into this protein drop, and because if the precipitate is formed instantly, then it is either the protein concentration is very high, or the precipitant what you have taken is also withdrawing the water very fast. So in either of these cases, you have to change the conditions. Since you see in these particular conditions, my drop is still clear, so this means both of these parameters are perfectly fine. Now I will keep this under in a crystallization incubator, and I will observe this plate after every 24 hours, and ideally I should see protein crystals by tomorrow morning. So this is all about the protein crystallizations. So with this, I would like to conclude my lecture here. Thank you.